Okay, so a very good morning and warm welcome to this innovation workshop, part of the Genesis Digital 2022 conference. So this morning's workshop is on the topic of important considerations for patenting biotechnology in Europe and the USA. So it is in collaboration with One Nucleus partner, Appleyard Lees. During this session, Edward Rainsford, Senior Associate at Appleyard Lees, will discuss the different approaches taken to patenting the science technology at the Europe Patent Office and the US Patent and Trademark Office. So Edward brings with him 12 plus years of experience in being a life science patent attorney. I will shortly hand to Edward to do his presentation this morning, but from me, just a couple of housekeeping points before we start. So if you are not presenting, then that would be wonderful if you could keep your microphones on mute and your cameras turned off. As you will have heard, I am recording the session. So the aim is that all of our innovation workshops will be available to view as on-demand content for the conference. So this will be available from Tuesday, the 13th of December, and they will be available on the app until the 5th of January. So that way you can recap on the content if you wish to. And of course, the chat box will be enabled. So as with all our workshops, we do like to keep these as interactive sessions. So during Edward's presentation, please feel free to use the chat box to drop your questions into, and it will be my job to pass those to Edward at the end. Or Edward, by all means, if you feel like stopping halfway through, if we see the questions coming through, then happy to go with that approach as well. So with that, Edward, I believe you'll be sharing your screen. So I'm going to hand the floor to you now. Thank you very much for joining us. Thanks. Over to you.
Thank you very much. It's great to be sort of starting off the innovation workshops for Genesis 2022. I'm going to share my screen and hope that that comes up fine. Start at the start. So yeah, I'm going to be talking about some of the things that you may need to consider when filing patent applications at the European Patent Office and the United States Patent and Trademark Office. I'm going to give a bit of a whistle-stop tour. There's a lot of different things that we could talk about. This isn't an exhaustive list, and there's a lot more that could be said on each topic. So if you do have any questions, I'm happy to answer them. And if you want to get in touch with me afterwards, then please do feel free to. We can discuss some of these things in more detail. So to start things off, why do the differences between the EPO and the USPTO matter? Well, for starters, patents are territorial, and each patent office sets its own rules as to what is and is not patentable. So you can imagine that differences between these rules can lead to different types of objections that may an application may face. So if the requirements of important territories such as Europe and the USA are not considered when drafting a patent application, it can lead to a lot of issues downline. So it's important to think about these as early as possible. And also the law in each territory is always evolving, and it's important to try and craft a patent application that's going to address all of the issues that you know about now, as well as trying to foresee what may be coming in the future in certain areas of law as they're developing over time. And I would say life sciences in particular is a very rapidly developing area of patent law. So to start with, what is and isn't patentable? Well, in order to get a patent application, all patent applications have to be novel and inventive. So the invention has to be new, and it has to not be obvious in light of what's gone before. But in life sciences, there are also some specific things that you can and can't patent. So in general terms, in Europe, we have Article 54.2, which sets out that the following things are not regarded as inventions. This is a broad list in this article, and covers things like discoveries, theories, and mathematical methods, aesthetic creations, which are covered mainly by copyright, such as, paintings, novels, etc. Schemes, rules, and methods, and performing a mental act, playing game, or doing business, and computer programs. But that's only as such, and there are actually means to protect computer programs, but that's an entirely different talk. So there's also the presentation of information. But what about life sciences, technology, and inventions? What rules specifically apply to them for things that are excluded? So we have to look at Article 53 EPC. So exceptions to patentability, European patents shall not be granted in respect of A, inventions, the commercial exploitation of which are contrary to ordre public or morality, such exploitation shall not be deemed to be contrary merely because it's prohibited by the law of contracting states. So such things may be transgenic animals that are going to suffer unnecessarily. So basically anything that may be against the morals of the European system. So B, you have plant and animal varieties, or essentially biological processes for the production of plants and animals, but the provision does not apply to microbial processes or products thereof. So there are other means within the European system for protecting plant varieties. So there's carved out in particular from what's patentable. So the last one, and the one that we're going to focus on a little bit more today, is methods for treatment of the human or animal body by surgery, therapy, diagnostic methods practiced on the human or animal body, and the provision does not apply to substances or compositions for use in these methods. So you can't patent a method of treatment, but you might be able to patent what you're using for the treatment. We'll go into more detail in that in a minute. So what's the situation in the USA? So Code 101 defines what is patentable, and it's whoever invents or discovers any new or useful process, machine, and composition of matter, or any subject and useful or any new or useful improvement thereof, and you may have a patent. But abstract ideas, laws of nature, and natural phenomenon are excluded from patentable subject matter. And this is defined by many cases which have set this out. So you can't have these because they're defined as being the basic tools for scientific and technological work. So the court has said that integration of an abstract idea or law of nature or a natural phenomenon into a practical application may be eligible. So what's excluded here is a purely abstract idea, a law of nature, or a natural phenomenon. If you can combine that with something that's not related to just those, what they call judicial exceptions, then it may be eligible for patent protection. So they say in a certain judgment, it was said, we must distinguish between patents that claim the building blocks of human ingenuity and those that integrate the building blocks into something more. So what else? This leads us from this to a three-part test. It's defined as part 1 and 2A and 2B. So 1 is the claim to a process, machine, manufacturing, composition, etc. So is it to something that could be patentable? If yes, is the claim directed to a law of nature, a natural phenomenon, a product of nature, or an abstract idea? And if it is there something in the claim, as I said before, that takes it to something that's significantly more than just the exception? And we'll get into some examples of this in a little while. So one example is for 2B, if it's an immunization step that integrates an abstract idea of data comparison into a specific process of immunizing that lowers that the patients will later develop a chronic immune-mediated disease. So basically, it's not just the abstract idea of comparing data, which is effectively a mental process. It's mathematics. It goes beyond that and therefore facilitates something that leads to an outcome and to a treatment. So there's also the written description and enablement in the US. So this code 112 states that the specification shall contain a written description of the invention, of a manner and process of making and using it in such a full and clear and concise way, in exact terms, that it enables the skilled person to make and use the same invention. And it also sets forth a best mode. So the important thing here is that the specification must teach the skilled person in the art how to make and use the full scope of the claimed invention without undue experimentation. So the description must be sufficient to show that the inventor actually is in possession of the invention. And it's generally described that broad claims need broad written description. So what does this really mean, and what's the difference between an enabling something and a written description requirement? So the best definition I could find was from a Harvard Law Review, which states that the enablement compels the patentee to teach the skilled person how to make and use the invention as broad as it is claimed without undue experimentation. So you're teaching how you make the invention and how you use it. What are you doing to create it and use that invention? The written description requires the patentee to describe the invention in sufficient detail to allow the person skilled in the art to recognize that the inventor actually invented what is claimed. So this is what have you done? What have you done in order to create this invention? And enablement is how do you actually make it and use it. Now, if you're still not entirely sure what the difference between these two things are, you're not entirely alone. A lot of USPTO examiners conflate these two things, and it can sometimes be a little tricky to straighten it out and show and demonstrate that you actually can have fulfilled both requirements. But again, we'll get into some examples of this a little later on. So what does that mean? So why do we care? So it probably all sounds quite straightforward. There are some rules, but what does it actually mean between the differences between the two patent offices? So there is a distinct difference in how things are interpreted between different jurisdictions, particularly Europe and the US, and particularly in life sciences. So for example, the requirements in Europe allow the patenting of nucleic acid and protein sequences that are found in nature, provided you have an industrial application. So if you have discovered a new biomarker, a new gene, and you can use that in diagnosis of a disease, then you could patent the exact sequence for the gene as it occurs in nature. That is not the case in the United States. You cannot patent nucleic acid and protein sequences in the format that they are found within nature, as that would fall into the exclusion of a product of nature. So it is, however, possible to patent either something that's been genetically modified or a synthetic sequence. You have to claim it as a cDNA, and ideally, perhaps something that's not just the sequence found in nature. So that's a lot more limiting than in Europe. And I would say when claiming nucleic acid and protein sequences, it's really essential that you do include either a cDNA sequence or set out quite clearly how the sequence has been modified from that sequence which is found in nature. So when drafting a patent application, you really need to think about both ways of how you might claim it. The European version, where you may well have the sequence as it's found in nature, and you can then claim that within Europe. And then in the US, either, as I say, how a sequence has been modified, or as it may be claimed in a synthetic version as a cDNA. So moving on, talking one specific area of patentability of stem cells. Now, looking at Europe, we have Rule 28C EPC, which says that European patents shall not be granted in respect of biotechnological inventions which concern the use of embryo, the destruction of embryos for industrial and commercial processes. So in another case, which is G206, it said that EPC, Rule 28C EPC, forbids the patenting of claims directed to there's inventions which, as described in the application at the time of filing, could be produced exclusively by a method which necessarily involves the destruction of human embryos from which said products derived, and that the destruction is even if this the destruction of embryos is not actually claimed in the patent application itself. So if stem cells can be derived at the filing date by means that doesn't involve the destruction of a human embryo, then it may be patentable. For example, nuclear reprogramming, where you take a somatic cell and roll it back to either a pluripotent or a totipotent state, or parthenogenesis, where you may take an oocyte and sort of chemically stimulate it to divide, and you end up with something that is akin to an embryo, but is not actually itself possible of forming human life. So the case G206, at the time when that particular patent was going through this case, it wasn't possible, well, sorry, I should say, at the time when the case was happening, it was possible to derive the cell line by not destruction of a human embryo. But at the time the application was filed, which was the critical point, it wasn't possible, and you had to destroy embryos in order to arrive at the cell line. So that was deemed to not be patentable because it was involving destruction of a human embryo at the filing date. Now, it therefore is critical in European applications relating to stem cells, where you may be able to derive them from destruction of an embryo, that you do also include an explanation as to a means of arriving at the stem cells from a process that doesn't involve the destruction of a human embryo. So, practical tips in terms of Europe, as I say, ensure that the patent application describes how you can arrive at the stem cells without the destruction of a human embryo, and potentially insert disclaimers into the application at the point of drafting it, which may be useful to disclaim the destruction of human embryos to arrive at the stem cells themselves. In the USA, we have to follow the patent eligibility tests, which I set out earlier. And to give a bit of an example as to how we do this, say that an applicant specification discloses differentiating stem cells into pacemaker cells for use of regenerating a damaged heart tissue. So in our example patent application, in nature, pacemaker cells can be identified via a protein marker P on the cell surface. The pacemaker cells contain genes that are capable of expressing marker Z, but in nature, these genes are not expressed, and they do not occur in pacemaker cells that have the marker on their surface. So in nature, marker Z is not expressed. So expression of marker Z exhibits increased efficiency of utilizing oxygen more than naturally occurring pacemaker cells. So isolation, though, of these man-made cells does not change them in any way. So let's look at some claim examples. Claim 1, an isolated man-made human pacemaker cell. This would not be allowable because it is simply a product of nature. The fact that it's man-made and that it's been isolated by human hand and then grown up and passaged doesn't actually change the cells at the end from what they were at the beginning. Therefore, it's still a product of nature, even though they've been isolated. So 2, an isolated man-made human pacemaker cell expressing marker Z. So, as I said before, marker Z is not a marker which is expressed in human pacemaker cells as they're found naturally. So because we've modified these cells to express this marker, then it would be allowable, because the cells themselves, although being the same cell, have been genetically modified to express a protein that they would not express in a natural setting. And also we have a population of human pacemaker cells. We're in the population, apologize because the right hand side of my screen is not visible to me. Population expressing 10 to 15% positive for marker Z and 85 to 90% positive for marker P. Again, that's allowable because you are expressing this marker Z, which is not found in nature. So I may have to. So I'm seemingly unable to move on to my next slide.
I can share the slide deck if that's easier, Edward. Just let me know.
I will close and reopen it, I think. There we go. And I've also managed to see the right-hand side of my screen now. So claim 4, a composition comprising a population of isolated man-made human pacemaker cells in a container. So they're not in a natural setting now, they're in a container. But again, just because it's in a generic container does not actually change the cells that were isolated originally. So in this instance, it would not be allowable because, again, they've been isolated, they've been placed in an environment of which, you know, they're not naturally found, but it doesn't actually change the cells themselves. So 5, a composition comprising a population of isolated man-made human pacemaker cells in a biocompatible 3-dimensional scaffold. And this would be allowable because now you're growing the cells on a structure, and that has the advantage that you can then use the cells grown on that structure to implant them and into a human heart, for example. So this is, despite the cells being what they were at the start, they're now in a form which is not naturally occurring. So you've taken it slightly beyond what it was originally. It's not just cells on in a flask, it's cells in a specific format for a specific purpose, and that would be allowable. Now, it should also be mentioned that, again, these claims would have to fill the requirements of novelty and inventive step. So whether or not that's actually inventive over growing any other type of cell type on a 3-dimensional scaffold would be very debatable, probably with the examiner. So tips for the US. Ensure the application contains something more than just a natural product. Either it is genetically modified in some way, or it's in some form, as we saw in the last claim, that's not naturally occurring. So this has to be more, as I said, than just human intervention. If nothing differs in the natural product after it's been passaged, for example, then it's just the same cells that are found in nature. So, and also it's important to provide sufficient evidence that the changes to the natural product are what provide the advantage of the invention, and not the cell as it was found in nature. So moving on to medical methods of treatment. So in Europe, we have to recall Article 53C, that methods of treatment of the human body or animal body by surgery or therapy and diagnostic methods practiced on the human body are excluded from patentability. However, it is possible to attain a patent for the substances and compositions used in those treatments. So substance or composition X for use as a medicament, or for indeed in if you have a known substance or composition, but you have discovered that you can use that for the treatment of a new disease not known previously, then you can also gain a further medical use for that substance or composition. And it has to be formatted in substance or composition X for use in the treatment of disease Y. So while directly you can't patent medical treatment, you can use the composition substances which may be used in those treatments. So the claims are therefore limited to the use of a substance or composition. Okay, so therapeutic methods in Europe. Therapeutic methods are concerned with bringing the body back to a normal or healthy state. And methods of doing that are not patentable. However, providing relief to discomfort is not necessarily a therapy. One example is if you're cooling an animal in hot weather, you're not curing it of a disease, you're just lessening it of discomfort. So you're not bringing the body back to a healthy state per se by curing some form of disease. And also methods relating to cosmetics may actually be patentable if they don't relate to a method of treatment for a disease. So when drafting a patent application, ensure that there's basis for removing the therapeutic steps from what may otherwise be patentable method. And you can either do this by a disclaimer, by disclaiming the certain steps, or by omitting the steps in general. So in the US, it's patentable in the US, but you have to apply the patentability test and you have to fulfill the usual requirements of novelty and inventive step. But the claims must provide the physical steps which make it more than just applying natural relationships, going back to what we were talking about previously. So in order to meet the written description and enablement requirements, it must be set out what makes the treatment effective. So you have to sort of state why you're going to be able to treat this disease using the steps that you are setting out in the patent application. I say that ensure the application has sufficient experimental evidence to support the scope of the claims. And that's very important in the US. As I say, written description enablement can be applied quite strictly in the United States. So it's important to show that you do have that evidence that if you're claiming a very broad claim, that you can support that across the whole scope of that claim. So surgical methods in Europe. So treatment by surgery is excluded. And it's a method of treatment of surgery for cosmetic purposes or for embryo transfer is excluded from patentability, as well as surgical treatments for therapeutic purposes. So this is slightly different from the methods of treatment, because if it has any surgical step in it, then it's excluded from subject matter, whether it's for cosmetic reasons or otherwise. But what is defined as surgery? Now, invasive techniques is for a routine character, carried out by non-medical professionals in a commercial environment, that they're not excluded from patentability, such as tattooing, piercing, hair removal, microabrasion, and things like that. It basically anything that is an invasive surgical procedure that has to be done by a medical professional it will not be patentable in Europe. So ensure that there's basis for how a claimed invention functions, e.g. a medical device which does not involve a surgical step. One example of this is pacemakers. There are two cases at the European Patent Office, one of which is allowable, and that describes how a pacemaker functions within itself, how it makes itself more efficient. And that just describes how the device itself works. Another which would not be patentable is if you're describing how it applies the electrical current to the human body. So how a device functions in and of itself may be patentable, but how that interacts with the human body may not be patentable. It may depend on a case-by-case basis. So surgical methods in the US are patentable, but there are safe harbor provisions which prevent you from suing doctors when they're carrying out life-saving operations and surgeries. But you may actually be able to take a case for infringement against somebody who provides that doctor with the means to infringe your patent. So the medical device, etc., that may be used in a surgery. So diagnostic methods. Diagnostic methods in Europe. So in order to be excluded from patentability, the claim must include the method steps for all of these. So 1, an examination phase, collecting data. 2, the comparison of these data with standard values. Finding of any significant deviation, i.e. symptoms during the comparison. And then 2, the attribution of the deviation to a particular clinical picture, i.e. making a decision on what you have to do based on that data. So therefore, if one of these steps is not performed on a human or animal body, then it's not excluded from patentability. So if you can show that you have a lab step that is involved in any of 1 to 4, then it may actually be possible to get a patent for a diagnostic method in Europe. So in the US, a biomarker for a disease and its use may be viewed as a natural phenomenon and deemed ineligible for patent protection. So diagnostic methods that include only the observation of natural relationships without a step that it goes beyond this observations will not be patentable subject matter. So in order to overcome this, it may be necessary to limit the claim to a diagnostic test in combination with a treatment step or the use of a non-conventional reagent such as new antibodies, etc. So again, this is just trying to take it away from, well, here's a biomarker, it's just a protein, and we're just detecting a protein, and that's it. So including that treatment step takes it beyond just detecting things that are already there in nature into something that a human being actually has to do. So ensure that the application contains more than just a natural product. And here's a bit of an example of that. So here's a claim. It's a treatment method, and I won't go into the specifics, but it was deemed unallowable because it is an abstract idea, because of the mathematical calculations of determining the ratio of C11 to C13 levels is a mental process and could be performed by a human mind. So we are detecting something, we're calculating a ratio, and using this ratio to by measuring the blood samples to diagnose this based on the ratio and identifying if someone is a non-responder phenotype. But that's deemed to be a mental act, as it hasn't gone beyond anything than detecting something that exists in nature and then performing some maths on it. But claim 2 is allowable because it is wherein the treatment is a non-steroidal agent capable of treating NAS-3. So this is allowable because now we're involving that treatment step. We're doing something beyond just detecting things that exist and then doing some mathematics on it. Based on doing that, we have a determination, an action step that follows on from it. So claim 2 is allowable because, as I say, it instructs a treatment. So moving on to antibodies. So antibody patents in Europe, there is a set now, relatively recently set out in the guidelines for examination at the EPO, which sets out what can be claimed with regards to antibodies. So importantly, most commonly now, a lot of antibody patents relate to the structure. So if it's an IgG, it should be defined by no fewer than the 6 sequences for the CDRs. and the claim will be objected under lack of clarity if it has less than six CDRs, because it's generally deemed that essential technical features are missing. So that's also true when we're claiming nucleic acid sequences for an antibody, as well as the amino acid sequences. So you have to generally show the at least 6 CDRs for it to be clear, deemed at least by the European Patent Office to be clear and patentable. So you can also do it in reference to a target antigen, and an antibody can be functionally defined by the antigen itself, provided that you have clearly defined what that antigen is. So you can have a claim to an antibody binding to X, or an anti-X antibody. And obviously in this instance, the antigen has to be new. So target antigen or further functional features. So for example, you could define it in terms of the binding affinity, neutralizing properties, induction of apoptosis, internalization of receptors, inhibition or activation of receptors. So, and E, functional and structural features. So it's possible to claim an antibody that's characterized by sequences of both the variable domains or the CDRs with less than 100% sequence identity when it's combined with some of those functional features that we just mentioned. So production process is possible, as I say, it's claimed either by the immunization protocol of a human, non-human animal, as well as the characterized DNA antigen, or by the specific cell line producing them. And you can also define it by the epitope, a set of specific amino acid sequences on the antigen, or lastly, hybridoma. And you can submit the hybridoma to an international depository and use that as basis for your antibody in the application. So further antibodies binding to known antigens at the European Patent Office are deemed to not have an inventive step unless there's a surprising technical effect. So, or there's no reasonable expectation of success. So generally speaking, unless there is something in particular that is setting your new antibody apart from known antibodies binding to the same antigen, it is likely to receive lack of inventive step objections. So examples of surprising technical effects may be that it has improved affinity, improved therapeutic activity, reduced toxicity, etc., etc. So unless you can show that your new antibody to a known target has something that sets it apart from known antibodies, you will run into issues at the European Patent Office. So in terms of tips, make sure that you have 6 CDRs, or if you can demonstrate that actually it combined with less than that, then you may actually not have to show that, but you do have to provide that evidence that that's possible. So define as many functional features as possible, provide as much data as possible, and make reference to any surprising technical effects, or why it's generally thought of as the state of the art, that you'd have no expectation that you'd actually succeed to make a new antibody to a particular target. And links the sequences and the structure of the antibodies to the function. So for example, ensure the examples provided in the application relate back to the key features of the claims and any technical effects. And lastly, if you are attempting to claim a new antibody to a known target, then comparative data between what's known and your new antibody can be very, very useful. So you just to demonstrate that you actually have this surprising technical effect over what's known and that you have higher binding affinity, then having that comparative analysis can be very, very helpful when arguing against inventive step objections from the examiners. So what's the situation in the USA? Well, Again, you can have it by structure in the USA as well, such as we were saying, providing the sequencing for 6 CDRs. But unlike in the EPO, at the USPTO, they have a principle of structural non-obviousness. The general thinking at the USPTO is that it's unpredictable when creating antibodies what they're going to be like in terms of these functional features, binding affinity, etc., just based on raising an antibody to a known target. So as far as the USPTO is concerned, if you have a new antibody and you can show the structure of the antibody, and even if it's to a known target, you may still actually be able to get a patent application for it. So that's quite a key difference between the European Patent Office and the USPTO. So antibodies directed to epitopes, though, face a much harder time at the USPTO than they do at the EPO, and that is because of the written description and enablement requirements. So if you're trying to claim a antibodies directed to a new target, new antigen or epitopes, then you're going to face a lot more challenging time in the US than you're going to in Europe. So the USPTO previously used to use what they called a newly characterized antigen test, as a functional antibody satisfies the written description requirements if the target antigen is sufficiently described, i.e. by its sequence, which is somewhat similar to the situation in Europe. If you can define what the antigen is, then you can therefore claim antibodies binding to that antigen. That's not really the case at the moment in the US, and there's been some very recent cases which have illustrated how they view things now. So Amgen versus Sanofi, the federal court found that the claims directed to an anti-PCSK9 antibodies defined only by the target epitope placed an excessive burden on the skilled person on the basis that they would have to screen millions of antibody candidates to determine which antibodies actually fell within the scope of the claim. So these were the two patents that were basically revoked by the Federal Circuit, and they relate to an isolated monoclonal antibody wherein, when bound to PCSK9, the monoclonal antibody binds to at least one of the following residues. And then you have your list of residues. And the second patent is, again, it's monoclonal antibody to the same antigen, and it lists some different epitopes. So these were revoked on the basis that there is millions of antibodies that could fall within the scope of these claims, and the skilled person would be under an undue burden to figure out which ones actually do bind versus those which might bind, and the scope of the claim is therefore not supported by the written description. And there's new guidance to the USPTO in light of this. It said, in view of the Amgen decision, adequate written description of a newly characterized antigen alone should not be considered as adequate written description of a claimed antibody to that newly characterized antigen. Basically, that they hadn't supported their scope of claim enough, given the written description, the examples that they actually had in the patent application to begin with. So therefore, the antibodies themselves need to be defined in the claims. So the patents themselves that we just described, there was 20 examples of antibodies and 3D structures for 2 of the antibodies. So Amgen has actually requested, and the US Supreme Court has accepted, a referral. Basically, Amgen's position is that it's impossible to satisfy the federal court standard of enablement, that you cannot define enough antibodies in your patent application to ever have a claim directed towards an antigen or epitopes themselves, because you just wouldn't be able to define all of the possible antibodies that could ever bind to it. So the question that was referred is to whether enablement is governed by the statutory requirement that the specification teaches those skilled in the art to make or use the claimed invention, Or, but in another way, alternately, is the standard that you have to identify and make all or nearly all embodiments of an invention without substantial time and effort, basically saying it's impossible to actually do it if it is the latter rather than the former. So just to give you a brief view of where that there was 2 patents, as I said, in the US. There is only 1 patent in Europe at the time, and it's granted, and it's to a similar claim, slightly different sequences, but it again is to the monoclonal antibody or fragment that binds to human PCSK9 and is neutralizing. So this is a good example of a allowable claim in Europe that is not allowable in the United States. And if you may feel that it's actually down to the differences here, here's another more recent case, which is Baxalta versus Genentech, which was thrown out at the District Court of Delaware for invalidity for lack of enablement. And these claims are pretty much very, very similar, and it's an isolated antibody or antibody fragment thereof that binds to factor 9 or factor 9A and increases the procoagulant activity of factor 9A. So this was revoked in the US and is allowable in Europe. So it's going to be very interesting to see what the Supreme Court decides actually is the standard that you have to apply for written description and enablement in the US, because otherwise it's going to be borderline impossible potentially to get patents to antibodies that are directed to a specific target rather than defined by their own structure. So that's just been a bit of a whistle-stop tour through some of the different considerations and the different areas and the different approaches taken in Europe and taken in the US. So just to summarize, there are many differences between the 2 systems, and I certainly haven't touched on all of them. There are a lot more and a lot more different areas which you could talk about, and a lot more I could probably say about each of those areas. So it is important to determine how these differences may impact your invention and drafting a patent application accordingly. So providing sufficient experimental evidence can be very, very key to ensuring a smoother ride to an allowable set of claims. And I would strongly recommend seeking the advice of a patent attorney and discussing as early as possible the countries you're interested in filing, because as soon as you have a clear idea of where you are interested in filing, then you can start putting together a strategy and including the different fallback positions within an application that may address some of the objections that you might face, and also consider as early as possible what those objections might be and how this might change the scope of protection from country to country, because you might find that you have a patent application and you want to file it in some instances, for example, in the US, but it may have a very rough ride in the US, and that's something to consider before filing, I would say, rather than after filing. And with that, I would take any questions.
Excellent. Thank you very much indeed, Edward. I found that absolutely fascinating, the level of detail there between the different systems and jurisdictions. So yes, there is time and 10 minutes left of the session. I invite the audience to use the chat box to ask any questions, or if you prefer to use the emotions, you can raise your hand and I will spot that in the participant list. We do have one question that has come through from Martin, so I'll just read that out for you. So RE patenting naturally occurring DNA sequences in Europe, is the patent linked to and restricted by the commercial use? Thanks for that question, Martin. Over to you, Edward.
No, not necessarily. You can get DNA sequences, just claims to the DNA sequences themselves. So you may have subclaims, either dependent claims or maybe different independent claims related to the commercial use, but you can claim the sequences themselves, provided obviously that you're actually they're novel and inventive, based on what you are going to be using them for. So you need to know why you're claiming them, but you don't necessarily need to limit them to the commercial use. I actually have a direct message from Daryl, who says, do you anticipate any patent filing changes in Europe once the IVDR goes live? I think probably not. There's nothing that's going to change in terms of how the European Patent Office approaches the in vitro diagnostics. Generally, there's nothing that I'm aware of in terms of how they're going to change their standards, but it may over time affect it in terms of case law and how these are the changes that are coming in the IVDR. I think that's probably something that may develop in case law, but I don't anticipate it changing how the European Patent Office goes about patenting diagnostic methods through prosecution. But as I say, things may change as you end up with case law, and that may filter back in the future, but probably nothing specifically and nothing imminently, I'd say.
Edward, if you could, I know you've just been through in very thorough detail the differences between the two jurisdictions, but if you could sum up sort of in a sort of a key take-home message, whether there's more challenges. It's a bit of a controversial question. Where would you face more sort of challenges in terms of pushing through your invention? Is it the EU? Is it the US? What's more permissive or permissible?
I think it ultimately depends on what your invention is. If you're, as I say, if you are wanting to patent something that involves, say, a method of surgery, then you're going to have a much easier time in the US than you are in Europe. But again, as I said on my last section, which was talking about antibody patenting, if you are trying to get a patent for a new antigen, you're going to have a much rougher time in the US at the moment than you are in Europe. That might change based on what the Supreme Court say, but at the present time, antibody claims directed or defined by the structure of the antigen, you're going to have a much more difficult time in the US. So it really does depend on what sort of invention you've got, and the different objections that are set out in different places. There's different philosophies in Europe than there are in the US, particularly surrounding things like claims to methods of treatment. As I say, you can certainly get a method of treatment in the US, no problem. You might, as I say, you wouldn't be able to sue a doctor for performing that method of treatment or surgery, but as I said, you could potentially go after a company for contributory infringement, for supplying them with the means to infringe your patent in the first place. But you can't do that in Europe, barring, as I say, some of the workarounds or some of the things that I've described already.
Thank you. And just as we were waiting to come onto the call, actually, you mentioned that there are some updates in regards to patenting antibodies in EU. So we've got a few moments if you wanted to provide any further detail on that.
Yeah, there's a case that's just been heard, and they just had the hearing at what's called the Enlarged Board of Appeal, and things get sort of sent up from the technical boards of appeal up to the Enlarged Board of Appeal when it's a really crucial point of law, and this is one involving antibodies. And what they did was they filed their patent application, and it had different antibodies in it, and they didn't define what the technical effect. What they ended up relying on was that there was synergy between different antibodies within those that they filed, and the technical effect was supported by what was called post-published evidence. So basically they said, well, these antibodies which we've described, they have synergy. Here's some experimental evidence, which wasn't in our patent application, that supports the fact that these had the claimed synergy and that they work together. So that was granted, but an opponent said, well, what you've potentially done here is you've kind of got a new invention combining what was in the patent application, the antibodies themselves, and actually supported it by evidence not in the patent application. And is that fair? Is it right that should the skilled person who views patent applications with regards to novelty and inventive step, etc., should they believe that what you've got is plausible based on what's written in the patent application, or should it be just that it's not implausible based on what's written in the patent application? So if it's not implausible, then you can say, well, here's some evidence and I can work that out later, and that should be able to support my patent application. Or should you just really be locked into what you wrote in the patent application itself? So that's going through the enlarged Board of Appeal at the moment, and it may change the standard of how much data you actually need in a patent application in order to support the scope of your claim, or whether you can add that later to say, well, here's some post-published evidence that we've created that shows that what our claim actually says it does actually does. So it's going through the enlarged board at the moment, and it's going to be interesting to see how that works. I think we may have new question, which is how do the key differences play out in, for example, the cell and gene therapy area? So that sort of feeds into some of the things I talked about in terms of in the US. Is it what is there that goes beyond a natural phenomena? If you have got something in the gene therapy space, you won't be able to claim, for example, the the original copy of the gene that you're trying to correct, for example. So if you can claim something, the gene therapy, for example, in an AAV construct, then that might be something that you can claim in the US. That's sort of taking it again beyond just claiming maybe the correct copy of a gene that you're then trying to put into a human being. So there are ways to claim things within the gene therapy space which make it possible to claim in the US and Europe. And I think I could probably put an entire hour's presentation together with regard to gene therapy, but I'm more than happy to go through it individually. If you have any questions, please do feel free to contact me. Happy to talk to anybody and dive into things in a much more detail than I could do in a brief overview like this.
Thank you again. Thank you so much, Edward. And thank you to the audience for logging in and for engaging with your questions. It really does highlight the importance of having an expert in this area when you're working with your new inventions, and of course with these new modalities and new types of modalities as therapeutics. It's more important than ever. So with that, just to reiterate that you can use the conference app to contact Edward with your follow-up questions if you'd wish to, or just to make contact and introduce yourself. Please utilize that. And then we'll be returning at 11:30 today with our next innovation workshop in collaboration with EY on the topic in today's disruptive healthcare environment, how will medtech transform? So thank you once again for joining. I hope you enjoyed the session, and I look forward to seeing you on future sessions. Thank you again, Edward. Thanks, everyone. Take care.
Thanks very much, everyone.
Bye.